So, you have a test tomorrow, and we need to go over these essays.
Do it.
The test is over. Are you filming?
Mm-hmm.
Yay. Test is over, chapter 16 through 20.
Is it
Do you have an essay sheet? If you did, you would see that. Describe the process of mitosis and the cell cycle in detail. Discuss everything that happens in each phase. Include labeled diagrams of each phase in your essay.
I'm thinking we should send one.
There you go. You wanted to send her an essay sheet?
Yeah, that might be better actually. Your mom? Yeah.
She likes biology essays?
Yeah, she wants to know about it. She wants me to keep her in the loop.
Okay. Tell her I said hey.
Oh,
We're videoing the class. You can send her a whole video.
Where do you find those videos?
YouTube. My tube. Go to Mr. Willis's Awesome Biology class. Mr. Willis's Awesome Biology textbook. They're all there. Just type that out on YouTube. So, here we have a big cell. And here's the nucleus of the cell. And we go through all the different phases of the cell cycle. You should have all this in your essay. G1, S, G2, M, and C. Y'all know this stuff?
Wait, is this going to go on YouTube tonight?
Heck yeah.
Is G1?
It's going to be on YouTube in an hour.
Okay.
That's how fast I upload. Question?
Which part is interface? Is that G1, S, and G2?
Great question. So we call G1S and G2 interphase. And what happens in interphase? Well, you should probably write a few sentences on each of these. That's a good way to start the essay. G1 is when the cell grows and gets bigger and does its normal thing. It takes in food, it makes proteins, it moves stuff across membranes, it transports vesicles around, it moves those little motor molecules along. It does everything it normally does. Normal cell's life and function, and the cell gets bigger and bigger as it grows. That's G1. Are you happy with G1?
Say that one more time.
All of that stuff?
No, it's fine.
The cell grows and does normal functions. Okay. That's what happens in G1. The cell gets bigger. Questions about G1? What happens in S phase? Copies its DNA. And we went over that whole process, how it copies its DNA. That's that picture here. Remember, DNA helicase comes in and unwinds the DNA. Y'all remember that? And then DNA primase comes in and puts RNA primers onto the DNA chain.
Is this the first essay?
Heck yeah, see it right here?
Yeah.
And then DNA polymerase comes in and puts on new nucleotides on each side. On this side, it puts them on continuously, and on the other side, it goes in little fragments called Okazaki fragments.
And that's
all chapter 20. This whole thing is chapter 20. What were you going to say, Joseph?
It's going away from going.
You can go toward the DNA helicase on the leading strand, but you have to go away from the DNA helicase on the lagging strand.
And that's when it's Okazaki.
That's when it forms these little fragments called Okazaki fragments.
Do we have to know what Okazaki fragment?
That's for an honors biology section. So some of you do. Jenny, you good?
I'm okay.
Was Okazaki Japanese?
He was. So that's all in the S phase. DNA replication happens in the S phase. DNA is copied, and now you have 2 copies. What happens in G2? Grows more. What else?
Don't they line up in the middle?
It has no, not yet. It has something to do with this coming up.
That's like the embryos disappear.
Nope. In G2, the cell makes proteins that are going to be used in mitosis.
That's all we need.
Gets ready for mitosis. It also copies the centrosomes. The centrosomes here, copy. Now there's 2 centrosomes. Why do we need 2 centrosomes?
So you go on the other side.
Yeah, you're going to go to the other side. Do it, Lachlan. So, that's G2. So you should start out this essay with a paragraph on G1S and G2. Those make up interphase. That's the longest part of the cell cycle. All this takes, in a quickly dividing cell, this takes 23 hours, and this takes an hour.
How long would it take for a long cell to divide? What kind of cell would take longer?
Some of your cells, your brain cells and your muscle cells, they aren't dividing hardly ever. Hardly ever. Maybe not even once in a lifetime. So they're real slow or almost stopped dividing. But your skin cells, they're multiplying real fast. Your cancer cells are the fastest multiplying. They multiply so fast they form big lumps, tumors. How we doing? Kylie. All right. Next is M phase. What happens in M phase? Well, M phase is made up of 4 different phases. Prophase, that's my phase because I'm pro. Metaphase. Anaphase, that's Anna's phase. And telophase. Anna, did you ever find an anaphase yesterday? Was that the best part of your day?
Yes.
So, I'll show you what happens here in all the phases. You want to see it?
Sure.
First of all, in prophase, the chromosomes coil up where you can see them. Before, they were all spread out in the nucleus. What's DNA all spread out in the nucleus called? Chromatin. But now in prophase, they coil up so you can see them. Is there any evidence here that the DNA has been copied? Do you see the evidence that the DNA has already been copied in S phase?
No.
No, you don't see any?
Yeah.
Yeah, I do. There's 2 TT. No, I don't see it.
Anyone? Is there evidence showing that the DNA has been copied?
Yes.
What's the evidence?
The sister chromatid.
Yeah, the sister chroma. Each one has a sister. That's exactly like it.
Wait, what phase is this?
This is prophase.
Okay.
Each chromosome has a sister chromatid. That's the evidence that the DNA was copied in S phase. Every chromosome has a copy of it. Okay? What else happens in prophase?
Centrosome. Go to opposite side.
Centrosomes move to opposite side of the cell. Nuclear membrane dissolves, disappears.
Wait, this is prophase?
Spindle fibers form and shoot out from the centrosomes and attach to the chromosomes. That's all in prophase. That's a big paragraph you'd have to write. And then you'd have to draw a picture that looks like this. And do you know how to label your picture? Do you know how to label a drawing?
I mean, what if that?
The picture would look like that if we drew it.
You draw this, and then here's how to label it. Centrosome. What would I label this?
Spindle fibers.
Spindle fibers. What would I label this? Chromosome. I could even label that. What would I label that?
Centromere.
Centromere. A lot of stuff you can label here. Could even label this. What's this?
Cell membrane.
Cell membrane.
Yeah.
Not a cell wall unless it's a plant cell. Now plant cells have cell walls. You could label it cell wall if it were a plant cell. So you have to draw and label your drawings. How do I know that you have to label your drawings? It says include labeled diagrams.
Would we need to draw 6 chromosomes?
No. You don't have to draw 6. Draw as many as you feel like. Okay? How we doing? Questions? Anyone have questions? That was prophase.
Hold on.
Can I erase my labels?
Wait a second.
Yes. No.
Okay.
Erasers? Erasing? Okay. What happens in metaphase? That's the next phase. What does?
The chromosomes.
Whoops. Didn't need to erase that. Chromosomes line up in the middle in metaphase. Lined up in the middle. There we go. Draw that, label it again.
Oh, so we have to do all the phases.
Heck yeah, you gotta do all the phases.
That's all. That's all.
Oh, I didn't see that. Okay.
Metaphase, they line up in the middle.
That's all we need to say for it.
I would say the chromosomes attach to the spindle fibers and move to the center of the cell. Do y'all know what this line is called?
The equator.
The metaphase plate. It is actually the equator of the cell, you're right.
What is it called?
The metaphase plate.
And line up in the metaphase plate.
They line up at the metaphase plate, which is at the equator of the cell. Jenny was right. Why are you thinking?
Metaphase plate.
Metaphase plate.
Metaphase plate.
Metaphase plate, Joe. Metaphase plate. What's next, Anna?
Anaphase.
Yeah. Anaphase, the chromosomes separate and move to opposite sides of the cell. They separate at the centromere. Pulled by the spindle fibers. That's how they move. They're pulled along by the fibers. The fibers shorten, pull those chromosomes. How we doing? Yes?
Okay, is the middle of the chromosome called the centromere?
Centromere, right there where they're joined.
Okay, thank you.
You're welcome. Another question? Thomas, how you doing over there?
Pretty good. For anaphase,
Pay attention here. Yeah.
will we just have to write that it's where the chromosomes separate and go towards opposite?
Yes.
sides?
Anaphase, the chromosomes separate and move to opposite sides, and then you got to draw a picture of it. Yes. Yes. And telophase. You ready for telophase? Chromosomes arrive at opposite sides of the cell. chromosomes arrive at opposite sides of the cell. What else happens?
New nuclear membrane.
New nuclear membrane forms on both sides around the chromosomes. The nucleolus reappears. Yeah, you could try that.
Chromosomes come together.
The chromosomes uncoil back into chromatin. These chromosomes come apart. You won't be able to see them anymore. They uncoil, and now we just have another nucleus. And that's the end of mitosis. Mitosis is the division of the nucleus. And now that we have two nuclei, we're done with mitosis. And the cell will start to pinch inward. Start to pinch inward.
This is in telophase?
Yeah. And what's it called when it finally breaks apart in two? What's that called? Cytokinesis is when it finally breaks in two. That's the C. And now we have two new cells. And we're back in G1. After C, it goes back around to G1. That's why it's called the cell cycle, because it starts all over.
So we're not gonna.
So what are these cells going to do next?
G1.
Get bigger.
They're going to get bigger. They're going to start growing. They're back in G1. Yes, Alex.
Is there any more information besides them splitting inside pieces?
Not really. That's all it does. Well, you can say they form cleavage furrows. Do you remember that? They form cleavage furrows. And you can say in plant cells, they can't form cleavage furrows. In plant cells, they have to build a new cell wall. That's called a cell plate. You could say all that. Yes, Yosef.
Are we not going to have to know, prophase 2, metaphase 2, anaphase 2?
That's meiosis, not mitosis. That's when we make sex cells.
So we don't have to write
And
about
remember that this has to separate again?
So we're not writing about that?
That's meiosis.
Oh, sweet.
No, you don't write about that in this essay. But you need to understand it for the test.
Yeah.
Because there's a whole chapter on that.
Is there?
Chapter 11, I think.
Word.
Avery, you good? Nice. Questions, anyone? That's the first essay. How many are going to write that essay?
You're good.
I hope.
You go have it.
Can I go to do what? Someone said, can I do something? I didn't hear it.
Oh, I said you have to go over the other one.
I'm going to go over the other one. You ready? Let's go over the other one. The other one is the problem sheet. The problem sheet. Take a look at your problem sheet. I'll go over all these problems with you. I just handed it back. Oh, you aren't here? What in the world? What in the wide world of sports is going on? Is it up front here? Ah. Anna Grace. There you go. All right, problem sheet. Problem sheet. Here it is. Oh. You want to do some problems?
Sure.
All right. You want genetics problems? I got genetics problems. So when you get the problem sheet on the test tomorrow, it'll look like this, and you'll have to do these problems. There might not be 11 of them. That's kind of a lot. There'll probably be more like 6 or 7 of them. You don't know which ones you're going to get. I'll put some of them on there, some of them I won't.
But they're from the sheet.
They're like the sheet, but they're not exactly the same. I change them a little bit so you just can't memorize. So for instance, this first one, a heterozygous plant with yellow peas, big Y, little y, is crossed with a homozygous recessive plant with green peas, little y, little y. What are the percentages of phenotypes of their offspring? So that's just simply a Punnett square, right? Big Y crossed with little y, and you get big Y, little y, little y, little y, big Y, little y, little y, little y. What are those two right there?
50-50.
What color are those? Blue?
Yellow.
Orange? Those are yellow. What color are these?
Green.
Those are green. So you could say a 1-to-1 ratio, 1 yellow to 1 green. Or you could say 50% yellow, 50% green. Y'all see how to do that? So what I might do on the test tomorrow, Erica, I might not give you big Y crossed with little y, little y. I might give you big Y crossed with big Y, little y instead. I'll change it up a little bit so you can't just memorize. See what I'm saying? Not me?
No.
Questions about that question? That was pretty easy. Let's go to number 2. A woman carrying cystic fibrosis marries a normal man. What is the chance that they will have a kid with cystic fibrosis? Cystic fibrosis is a recessive disorder. So, what is the woman if she's carrying cystic fibrosis?
Big Y, little y, or little y, little y.
Let's use C.
Little c.
No, she's
Big
big
C,
C,
little
little c. She's a carrier. The carrier doesn't have it, they're just carrying it. So she's got, this is the normal gene, that's the cystic fibrosis gene. That's her. And she marries a normal man. What is he?
Big C, big C.
Nope.
Little c.
Oh yeah, no, you're right. He's big C, big C. Sorry, Joe, you were right.
Okay.
That's the cross. So what are the kids going to be?
No.
Kids are going to be no?
0% chance.
No.
You could get big C, you could get big C, little c, you could get big C, you get big C, little c. None of the kids are going to have it. So there's a 0% chance that the kids have hemophilia.
Cystic fibrosis.
Oh, cystic fibrosis, thank you.
Is hemophilia in the hemoglobin?
Hemophilia is, yeah, it's a problem with blood clotting. So
Which deals with hemoglobin.
hemoglobin is found in blood, so they're related. 2 carriers for Tay-Sachs disease get married. What is Tay-Sachs? Oh, Tay-Sachs disease is a recessive disorder. So what are the 2 carriers for Tay-Sachs? What's their genotype?
T, little t. Big T, little t, big T.
What?
You have a big T, little t, cross with a big T, little t. Y'all know how to do that one? Those are your 2 carriers for Tay-Sachs. And you can check your work on this. By the way, you'll want to check your answers here. When I graded these, I did it kind of quickly. And I looked at the Punnett square to make sure you had it set up right. But I may have missed it. So check your answers and make sure that you got the right answers here. What is this? What percentage of the kids?
25%
25 what?
will have it.
25% will have what?
Cystic fibrosis. Tay-Sachs.
25% have Tay-Sachs. 75% don't have Tay-Sachs. 50% are carriers for Tay-Sachs.
Don't you get tested?
Don't know what you said.
Don't you get tested before? Sometimes, like if.
You could
You can
see if
be
you're a
tested.
carrier. Yeah, there are tests for it. What about Huntington's disease? A woman heterozygous for Huntington's disease marries a man without Huntington's. What chance does a child of theirs have for Huntington's? Huntington's disease is a dominant disorder. What's it mean if it's a dominant disorder?
Big.
Big H, yeah, it's big H if it's a dominant disorder. So here's the woman. She's Huntington's. She's got it. She marries a man without it. What is he?
Little H, little H.
Little h, little a. Half their kids are going to have it. Half are not going to have it.
Do you not need to have two big H's to have it?
Nope, you just need one. That's what a dominant disorder is. In a dominant disorder, you just have to have one gene. Both of those have it. Have Huntington's. Questions or problems?
Wait, so
Alex.
Can you explain the difference between when it asks the genotype and the phenotype?
Yes, genotype are the letters.
Okay.
Phenotype is what it looks like. Tall, short, has the disease, does not, type A, type B. If you're talking about the letters, that's the genotype. Yes?
so anytime there's big H in that, it has Huntington's.
It has Huntington's. That's because it's a dominant disorder.
But isn't Huntington's like just for girls?
Nope. Guys and girls can get Huntington's. What about snapdragons? They show incomplete dominance. If a pink snapdragon is mated with a white snapdragon, what will be the percent for the next generation? So in snapdragons, big R is red, little r is white. If you're big R, what are you?
Red.
Red. If you're big R, little r, what are you?
Red.
White. Red.
Pink. That's what incomplete dominance is. If you're little r, what are you?
White.
White. Incomplete dominance is when the heterozygote is pink or has half of the of each gene, part of each gene in it.
That's called incomplete dominance.
Incomplete dominance. See, I said right there, if a pink snapdragon, big R, little r, it's pink. So if a big R, little r is mated to a little r, what are the kids going to be? Big R, little r, what's that?
That's
Pink.
Pink. What's that?
pink.
White.
Pink. What's that?
White.
White.
White. What's that?
White.
Half white, half pink. That's the answer for that one. Erica, did you get that one right?
Yes, it is.
Good job. Number 6 is about blood types. A man with type AB blood marries a woman with type AB blood. Do y'all remember all the genes for blood types?
Yes.
IA equals type A. IB equals type B. Little I equals type O. So, if the man is AB and the woman is AB, what could the kids be?
Oh.
Wait, is that what it says? Yeah. Could you have a type A kid?
50%.
That's a type A kid. This kid is type AB. This kid is type AB. And this kid is type B. Type A, type AB, type AB, type B. What are the chances of getting a type O kid out of that marriage?
0.
0. If they have a type O kid, something's wrong.
You are not the father.
Not only are you not the father, but she's not the mother. Somebody switched babies with you.
Switch at birth.
Switch at birth. We're running out of time here, so I'm going to skip 7, but that's how to do blood type.
How do you do sex linked? Can you do?
Let's do sex link. Let's do number 8. What were you going to ask? Can I do?
Number 9 and 10.
Yeah, let's do 8, 9, 10. So, if the problem ever asks for sex linked, Grace, you with me back there? The problem ever says it's sex linked, that means you have to use X's and Y's. XX is female, XY is male. So both 9 and 10 say that it's sex linked. A lot of y'all missed this. See how it says sex linked there? Both 9 and 10 refer to sex linked traits. That means you have to use X's and Y's. So, for what? Hemophilia or color blindness, this is a normal female, that's a carrier female, that's a colorblind female. There's a normal male, there's a colorblind male. So what do we want to mate here? Colorblind male mates with a carrier female. Colorblind male mates with a carrier female. That's this and this mating. What if that mated with that? Well, now your Punnett square has to have X, XC mated with XC, Y. And so you're going to get X, XC, 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 XY, XCY. Carrier female, colorblind female, normal male, colorblind male. 50% of the kids will be colorblind.
What if they only had one kid?
Then it has a 1 in 4 chance. 1 in 4 chance of being that, 1 in 4 chance of being that, 1 in 4 chance of being that, 1 in 4 chance of being that. Are you confused?
No.
So, let's look at number 9. This is another sex link. A woman carrier for hemophilia. What is she?
XX, little x. Little x, little x.
She's this. She's a carrier. Instead of C, it's H. She's a carrier for hemophilia. And who does she marry?
Normal.
A normal man. Can you do a Punnett square with those?
Sure.
Those of y'all that missed this question?
Wait, is this number 9 or 8?
This is number 9. A woman carrier for hemophilia, XXH, marries a normal man, XY. What are the kids going to be? What is that?
That's
Normal female. What is that? Carrier female. Good. Somebody say it right there. What's that? Normal male. What's that?
diagnosed male.
Hemophilic male.
So what would the percentages be?
None of the females have it. 50% of the males have it. Or you could say 25% of the kids have it. Ten is kind of done the same way. You got to use X's and Y's. So when you see sex link, you got to use X's and Y's. Yeah.
Will the question like number 11 be on the...
Yeah, pedigree. Do y'all know how to do pedigrees?
No.
No.
Circle is female, square is male. If you color them in, they're affected. So, if I'm going to draw this chart on the left, I'll copy the chart on the left for you. Square, circle, square, circle, square. Look, I'm copying the chart on the left. Circle, circle, circle. That one's affected. That one's affected.
Will it give you the pedigree?
Heck yeah, they'll give you the pedigree. Now we got to figure out what everything is. We know these are little d, because they tell us that in the problem. So far, so good? Stop me if you have a question about pedigrees. Now, what else?
2 would have to be.
All the other ones have to have a big D.
Yeah.
Every single one of them. Because it's not colored in. So you know they have one big D. Now we got to figure out the other. How do you figure out what the other one is?
Punnett score.
You look at the kids, you look at the parents. Here's a parent that's little d. Don't they have to give one of those genes to their kids?
Mm-hmm.
Every one of these kids had to get a gene from this parent. So that's big D, little d, that's big D, little d, that's big D, little d. Yes, Tom.
The circle, like the parent, would be big D, big D.
This one?
Yes.
It could be big D.
Question mark.
But couldn't it be big D, little d?
No.
No?
Yes.
Because the kids are affected.
This kid is big D, little d. Couldn't it have gotten the big D from this parent and the little d from that parent?
Mm-hmm.
This kid is big D, little d. Couldn't it get the big D from there and the little d from there? Couldn't this kid get the big D from there and the little d from there? This could be big D, little d. It could also be big D, big D.
Is it D question mark?
It's a D question mark.
So you could say either one, it would be right.
Now you got to say it's either.
So,
You can't be for sure that it's that one or that one. So it's big D question mark. That's the right answer. Yeah. Yes.
if you put big D, would you be wrong?
Yeah, technically you would. It's probably big D because all three got the big D from that parent, so it's probably big D. But it might be big D, little d. We don't really know. What about this parent? Do we know what that is?
Little D, big D, little D?
Is it big D, little d?
Yeah.
Why? How do we know that?
Because one of the kids is.
It had a kid that's little d, little d. It had to give that kid a little d, therefore that's little d. Yes?
How do you know that all the kids in the middle are big D, little D?
Because they have a little d parent. So they had to get a little d from that parent.
Every time?
Yeah. This parent gives one of its two genes to its kid. Every time. You know, if all you have is little d, then the kid has to have it.
Oh, but none of them are colored in, so they have to be. Okay.
Now you got it.
So if it's not colored in, it has to be dominant.
If it's not colored in, it's got to have at least one big D. What about this kid?
Question mark.
It got a big D from one parent. It could have got either from the other parent. It's big D question mark. It doesn't have any kids, so there's no way to know. Are you good? Okay, so here's we go. You can get 30 points, you and your partner here. I'm making a Punnett square. Fill out what every organism is in terms of big D and little d. If you get the whole thing right, you get 30.
Do we need, does it matter male and female? Not really.
You aren't using X's and Y's here. You're using big D's and little d's, just like these Punnett squares.
Your knowledge of
And this will decide who wins the game. Green has 385 right now. 375 for purple. 395 for peach. It's a close game. 500 for orange. They're running away with it.
Oh, it's the same thing. Is it with D's?
505. No, they aren't running away with it. 505 for blue.
Which rule?
Yes.
Could I send you the essay tonight?
Do what?
Can I send you the essay tonight?
That's good.
All these have to have a description on the end of it, right?
Are you gonna be here during show?
Fill it out.
But then if this one has that one, then it has
Fill it out.
this one has it, so these big little things. Same with this one.
Ask your partners. Jenny, you're not helping with your partners? They're filling out Punnett squares. You have to hand this in even if you don't know it. What color are you?
Oh, we're not finished.
Write your color on your sheet. Look at green. Green. Got them all right. 30 points. They got a 415.
Okay, let's go for it.
Team Orange. Team Orange got 2 wrong. They get 10 points. Team Peach. Team Peach got 2 wrong. They get 10 points. Team Purple. They got 2 wrong. They get 10 points. Team White got 2 wrong. They get 10 points. Anyone else? I'm going to show you the answer. Sorry, we're out of time. Here's the answer. Both of these. Listen, have a seat.
Can I give you my paper?
This one's, look at the answer. This one's little d, little d. Its parents have to be big D, little d. We know all of them have big D's. Their kids are both big D, little d, because this parent was little d, little d. So those kids are big D, little d. These got a big D from one, you don't know from the other. Both of those are question marks. This parent, you don't know what they have. That's a question mark. That's the answer.
We have our look.
It's too late. Too late.